Have you ever seriously wondered if we could build a functioning piece of a human brain in a lab? I don't mean just a blob of cells, but a real working circuit. Well, you're going to want to stick around for this because what used to be pure science fiction is, well, it's becoming scientific fact. We are talking about literally architecting a synthetic brain.
You say, for the longest time, the general thinking was that lab-grown brain models were, frankly, just disorganized clumps of cells. Interesting, sure, but not really functional. But imagine a breakthrough, let's say in 2025, that just completely shatters that old idea. One that proves these models can be so much more. That's exactly where we're heading. So to really wrap your head around how massive this leap is, we've got to look back at the old way of doing things. For years, we were kind of stuck, looking at the brain through a pretty limited lens. Think about it like this, right? The old models, things like 2D cultures or even a single brain organoid, they were like having just one piece of a computer, like having the most powerful processor in the world. It's impressive, but by itself, it can't do anything. It can't run a program, it can't connect to the internet, it's just an isolated chip. And that was the exact problem scientists had when they wanted to study complex things like memory or disease. But that all changed. The huge breakthrough wasn't about growing a better single piece. It was about figuring out how to connect all the pieces together. And let's just anchor this whole paradigm shift to the year 2025. Imagine a research paper dropping that just fundamentally rewrites the rulebook for how we model the brain in a lab. You can just feel the shockwaves from a discovery like that. Okay, so the core idea, the secret sauce here, is fusion. Researchers figured out how to take separate, specialized mini brains, these are called organoids, and physically put them together. And what happened? They grew into each other, creating a single, unified circuit where neurons from different parts were all talking to each other. Suddenly, you had this complex, coordinated activity just emerging on its own. And this is where that computer analogy really clicks. If the old way was just having a single chip, this new way is like building the entire motherboard. You're connecting the CPU to the RAM to the graphics card, and all of a sudden, you don't just have parts anymore, you have a functional system. The whole thing becomes way more than just the sum of its parts. So, how in the world do they actually do it? Well, it's not magic, it's a really precise 4-step bioengineering recipe. Let's break down exactly how you build one of these circuits, pillar by pillar. Everything rests on these 4 pillars. First, differentiation, to make the parts. Then, fusion, to connect them. After that, maturation, to let the whole thing grow up a bit. And finally, interrogation, to see if it actually works. All right, pillar 1, differentiation. Here, scientists take stem cells and basically nudge them with chemical signals to become different kinds of brain tissue. It's how they can grow these distinct little organoids that mimic specific brain regions, like the cortex for thinking or the hippocampus for memory. These are your individual computer chips, ready for assembly. Next up is the absolutely crucial step, fusion. They take the different organoids and place them right next to each other in a special gel. And then, with a little encouragement, the neurons from one mini brain start reaching out their little tendrils, their axons and dendrites, across the border, literally wiring themselves into the other. This is the moment the motherboard gets built. It's incredible. Now, once it's fused, the circuit isn't ready to go right away. It needs time to mature. We're talking months. And over that time, those new connections get stronger, and the network starts to fire in these coordinated, synchronized bursts. That's a huge sign of real, living brain tissue. It's basically booting up and learning to run its own operating system. But how do you know it's really working? I mean, really? That's the final pillar, interrogation. Scientists use these amazing tools, like microelectrode arrays, to listen in on the network's electrical chatter. And even crazier, they can use something called optogenetics, which lets them activate specific neurons with a pulse of light to write signals into the circuit and see how it reacts. It's the ultimate diagnostic test to confirm, yes, we've built a functional system. And building this thing isn't just a cool science experiment for the sake of it. No, this marks the beginning of a completely new era for how we understand the human brain. This slide just lays it all out, doesn't it? The contrast is staggering. We've gone from looking at isolated parts to studying a whole interconnected system. We've moved beyond just relying on animal models to actually studying human-specific biology. And we've jumped from looking at a static snapshot of cells to watching dynamic, real-time network activity. It's a total game changer. So why should you care?
Well, this technology unlocks possibilities that were just dreams a few years ago. We can now test new drugs for diseases like Alzheimer's on actual functional human brain circuits. We could grow a personalized model of a patient's own brain to figure out the best treatment for them. We even have a brand new playground to start exploring the deepest mysteries of all, like consciousness and thought itself.
This whole thing really pushes us to the very edge of what we thought was possible. It forces us to think about the future of the mind, both biological and, well, technological. When you boil it all down, this is the fundamental shift. For 100 years, neuroscience has been amazing at breaking the brain down into its tiniest parts, the cells, the molecules. But now, for the first time ever, we have a tool to start putting all those pieces back together and watch the system actually come to life. And that leaves us with a pretty profound question to chew on, doesn't it? As our ability to architect these synthetic brain circuits gets better and better, we have to ask ourselves, where does the biology end and the technology begin? That line is getting blurrier than ever.